cell cycle and mitosis. Billions of cells die in the human body every day. To make up for this, our body produces millions of new ones. These cells are made from already existing cells. Most of the cells in our body divides except the few ones like cells present in your heart, the cells present in your muscles, and cells present in your brain. These are known as permanent cells. Rest of the cells are characterized into two types, that is, stable cells and liable cells. Stable cells don't divide all the time, but they are capable of division if it is required. The liver cells in our body are best example for stable cells. Liable cells includes cells which are constantly dividing, that is, cells in our mouth, intestines, etc. Cell cycle. A cell cycle is a circular path in which the cell grows and copies its genetic material and divides into two daughter cells. These daughter cells undergo the same cell cycle to produce more new cells. The cell cycle is basically divided into two phases. They are interphase and mitotic phase. The interphase and mitotic phase are further divided into many phases, that is, the interphase is divided into three phases, G1, S and G2. The mitotic phase or the M phase is divided into two phases. They are mitosis or karyokinesis and cytokinesis. Interphase. The cell spends its maximum time in interphase. During this phase, the cell obtains nutrients, metabolizes them, grows in size and duplicates its DNA, etc. The cell does many other steps all in preparation for the actual cell division. During the G1 phase of the interphase, cell increases in size and functions normally. New cell organelles are produced. Increases protein synthesis. After the G1 phase, the cell can decide whether it wants to divide or not. If the cell senses that cell division is not required, it passes into the G0 phase, during which the cell remains silent. If the cell decides to go on cell division, it passes into the next stage of interphase known as S phase. The S phase or synthesis phase is associated with the synthesis of new DNA from the old one. The cell doubles the number of chromosomes for the synthesis of new DNA. The next phase is known as G2 phase, in which the cell continues its growth and gets ready for the cell division. The mitochondria of the cell doubles and the cell continues to grow until the process of division starts. The next stage in the cell cycle is the M phase, which is the actual division phase. M phase or mitotic phase is divided into two major phases. They are mitosis or karyokinesis and cytokinesis. The cells in the M phase has completed the interphase and are ready for division. The first part of the M phase is mitosis, which is a step in which the nucleus of the cell divides into two equal and new nuclei. This happens before the cell actually divides, so the time after mitosis, the cell has two nuclei. Mitosis is divided into four major stages. They are prophase, metaphase, anaphase, and telophase. During the interphase, the DNA of the cell present in the nucleus is folded like a long coiled loop. This arrangement of DNA is unstable. This could lead to problems during cell division. So, during the prophase, the DNA condenses to X-shaped structure known as chromosomes, which can be easily seen with the help of microscope. This process imparts stability to the DNA. During the metaphase, the membrane around the nucleus disappears and the chromosomes lie free in the cytoplasm, and later these chromosomes line up at the center of the cell or the equator of the cell. One important cell organelle which help this process after this is the centrosomes and the microtubules projecting from them. Together, these centrosome and microtubules are known as mitotic spindle. The next phase is known as the anaphase. The centrosome starts pulling the chromosomes apart to the opposite poles of the cell. Since the chromosomes were duplicated in the S phase, each pole of the cell receives a full set of chromosomes. The next phase, known as the telophase, a membrane forms around each set of chromosomes to create two new nuclei. This completes the process of nuclear division, also known as mitosis. The next phase we have is cytokinesis. Cyto means cell and kinesis means movement. In this phase, the single cell divides in the middle and form two separate daughter cells, each containing a full set of chromosomes with the nucleus. To make the concept clear, let's watch a video.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
Hence, mitosis is the way of making more cells that are genetically the same as the parent cell. It plays an important role in the development of embryos, and it is important for the growth and development of our bodies as well. Mitosis produces new cells and replaces cells that are old, lost or damaged. From this video, I hope the concept about cell cycle and mitosis is clear. Thank you.